All right, welcome back, my dear students. Again, this is Aziz. I am not your teacher. I am just an adult who can help you with answers as for or as to how to learn better. I believe that the real teacher is inside you, not outside. Okay, I've always taught myself things, and I'm not advocating against teachers. As a matter of fact, I am a teacher, but and at this point in time in my life, I realized that if I can help students teach themselves, if I can eliminate myself from teaching my students, I guess I will have succeeded. So let's get started, okay? This is a continuation of our previous slide or series in regard to genetics. Excuse me. I'm going to share a page from genetics, a Regents Prep Exam page. We're going to start off with number 39 because last time I think we kind of got stuck on 39 with the word polypeptide. We're into polymers and other things, but then you got the point, okay? Sometimes I do get off. I do stray off track, so I apologize. You can always pause, fast forward, go back, or do whatever you want to do. Okay, I'm just gonna be me. Okay, I will just talk and talk and talk as if, well, again, I don't know anything, so I'm going to be teaching myself, and you just watch me how I teach myself. Okay, great. So let's do this. I'm going to. Here we are on page, let's see, page 8, statement or question or problem 39. A sequence of 3 nitrogenous bases in a messenger RNA molecule is known as. Now, from our previous lesson, we do know what nitrogenous bases are. There are 5 of them: A, T, U, C, G. However, when you see the word messenger RNA, it literally tells you do not talk about T, because our messenger RNA deals only with 4 nitrogenous bases, such as A, U. Well, actually, I'm wrong, and correct me, and you should correct me, because there is T. A, in mRNA, A attracts U, but there is T that would attract A. So there are 5 nitrogenous bases. Am I right? On both sides. Um, well, I don't want to say sides, but in DNA as well as in a messenger RNA molecule. In this case, we just need 3 of them. A sequence of 3 bases is called what? Polypeptide? No, we've gone that road, and we realized that was not it. A codon? Yes, it is a codon. Gene? No. Nucleotide? Now, remember the difference between nucleotide and nucleoside. Nucleoside, just so that you can refresh your memory, is something that has the nitrogenous base, sugar, but no phosphate group. Nucleotide is exactly the same thing with the phosphate group. Okay? So moving on. Question number 40. Which base is normally used in the synthesis of RNA, but not in the synthesis of DNA? Now, I kind of slipped in the beginning when I said uracil, or U, letter U, is not present in DNA. Remember that? So, but U is definitely present in RNA. So that kind of answers our question if you think about it. Adenine, that stands for A. Cytosine stands for C. Guanine stands for D, and uracil stands for U, which is only found in RNA. So which base is normally used in the synthesis of RNA, but not in the synthesis of DNA? B is the correct answer. Moving on. Base your answers to questions 41 through 43 on the diagram below of a biochemical process and on your knowledge of biology. Okay. Uh, just let's just absorb all this. I have no clue what's going on. There are a bunch of crosses and squares and parallelograms, but we got to do this, right? So we have to understand what's going on here. Now, glycine. I see glycine. I see leucine. I see a bunch of amino acids. Actually, if you think about it, let's read number 41. The synthesis of structure S. I'm sorry. The synthesis of structure X occurred in the where? Synthesis of structure X. Where is X? Well, what X is going to be, I'm going to give one of my, let's see, I'm going to get taken here. Oh, there you go. I am already taking. Oops, not so quickly, but anyway, I'm so sorry. I should not have done that, but I'm definitely trying to show you X is right there. The synthesis of structure X occurred in the where? Nucleus, cytoplasm, lysosome or lysosome, or vacuole. Now, vacuole, we know it does not happen there. Lysosome, we know it does not happen there. Nucleus, we know the synthesis happens. However, in the nucleus, you're not going to synthesize. Hold on. Let's take a look again. And remember, I am learning as you and I go together. You know, I'm learning together with you. So don't forget that. And don't feel embarrassed, or because I'm not. I don't feel embarrassed, and I don't know this. Okay, and I am doing it as we go with you. Threonine and arginine. Okay, let's think. UGUACA. All right, A attracted U, we know that. C attracted G, we know that too. And I am talking about right here, this relationship. A attracted U. Now, the U part is the RNA. The ACA part, that base is DNA. So when this happens, when you see DNA meeting up or not meeting up, but in the same place with RNA, it's, in my opinion, it's definitely nucleus. Because if you have DNA, well, just because it's, hold on a second, I do see uracil right here as well. Look at this, uracil, right? Which means we have uracil on both sides. This is not in the nucleus. I think the right answer is B, cytoplasm. What do you think? Am I right if I say it's B, cytoplasm? Let's take a look at the statement again. 41, it says the synthesis or the formation, right, from smaller parts into a larger part of structure A occurred. Now, if you pay attention to this, it says occurred, not ended up, so to say. Occurred the synthesis, so the beginning, Okay, let's paraphrase this. This strand started as that strand. Where? Where did it start? Where did it occur? That is the question. That strand could be floating inside a plasm. We know that. But that's where it ends up. But where does it occur? Where does it start? The synthesis of it. That answer is nucleus. Okay, moving on. 42. Which amino acid would be transferred to the position of codon CAC? All right. CAC basically says match, right? So what can we get? If C attracts G, A attracts U, and then C attracts G, that is going to be GUG. Do we have that? GUG, histidine. Do you see that? For those of you who don't see that, it is right here. Let's make it larger. It's right here, and it's right here. So that makes our answer D as in David correct, 42D. Moving on. 43. The biochemical process represented in the diagram is most closely associated with the cell organelle is known as the, okay, honestly, I don't know why, but I had this, I spaced out, okay? So let's go back. The biochemical process represented in the diagram, which diagram? I don't even have the diagram anymore in front of me, which actually I do, it's right there. Again, I'm spacing out for some reason. I just ate, probably, maybe that's why. But the biochemical process presented in diagram is most closely associated with the cell organelle. Cell organelle. Okay, so focus, Aziz, focus. You're preparing for Regents prep in a living environment. You gotta focus, right? So let's do this. It says organelle, okay? And then it says diagram, okay? Most closely associated with that organelle. Let's see, mitochondrion. Well, first of all, that's not an organelle. These are organelle. Well, actually, it is an organelle because mitochondria would be plural. Mitochondrion is a singular organelle, the powerhouse of the cell. So, but no, I don't like that because we don't have anything of power going on here, right? So no mitochondrion, chloroplast. I don't know what that has to do with any of this right now. No, ribosome. I like that. I like this organelle, and I call it a machine, a device sometimes. I definitely like this answer. Nucleolus. What is nucleolus, by the way? I really want to find out. Even though I know the answer to 43, it's ribosome, I definitely want to find out what nucleolus is. So shall we go and actually check this out? Let's see. Nucleolus, right? Not nucleus, but nucleolus. I'm sure I misspelled it. There you go. Nucleolus, a small, dense, spherical structure in the nucleus of a cell during interphase. Well, sounds like it's a nucleus within nucleus. There, uh-oh, I clicked on something too fast. Look at this. Nucleolus is the nucleus of the nucleus. I like that. Okay, now that I know what that looks like, I'm going to go back to my notes and let's see. That's it. I'm done. I'm done with this page. So we're going to move on to Unit 5, Genetics, number 44. The diagram below shows some of the DNA in a bacterium into which a human gene X has been successfully inserted. Okay, DNA of bacterium, we see that. That's probably some sort of enzyme, and that's what it's called. Um, and then cell wall of bacterium, okay. The bacteria that result from reproduction of the cell will most likely have the ability to, the bacteria that result from reproduction will have the ability to replicate all of its genetic instructions found in humans. All of the genetic instructions? Okay, that's interesting. Um, why would it replicate all of the genetic instructions found in humans? That's strange. B, produce vaccines to be used to immunize humans. Okay, I don't see anything that talks about vaccines here. Produce a human blood cell according to instructions in gene X. We don't know if gene X is a human blood cell or if it does contain human blood cell instructions. D, produce the human protein coded for by gene X. I think that is our winner. Number D. Again, I don't understand A, B, C so much because they're kind of irrelevant. D definitely talks about production of the human protein coded for by gene X. Remember, I think you remember one of those questions about insulin, where genetic material coded for insulin would be inserted into bacteria enzyme so that that bacteria would produce insulin, the human compatible insulin. Okay? 45, the diagram below represents the chemical pathway of a process in a human liver cell. Okay, so we have substance A going to, turning into or becoming substance B through a process of enzyme X using up the energy, and then substance B turns into substance C by virtue of enzyme Y along with the energy. So a particular liver cell is unable to make substance C. One possible explanation for the inability of the cell to make substance C is that um, right off the bat ran out of energy. But do we have that? Let's see, access energy for step 2 prevented the conversion. I don't think that would be the case. An excess of enzyme X was present. An excess, what's up with this excess? I don't understand. Was present, resulting in a decrease in the production of substance B. Okay, I don't understand that. C, nuclear DNA was altered, resulting in the cell being unable to make enzyme Y. Bingo, I think that's our right answer because you need enzyme Y to be converted or turned into substance C. Let's hold on that. I'm sorry, D as in David. A mutation occurred, which is kind of the same as C, nuclear DNA being altered, causing a change in the ability of the cell to use substance C. That makes no sense. The beginning was kind of right, but the end is not. So C, I would go with the C as our correct answer because nuclear DNA was altered, resulting in the cell being unable to make enzyme Y. In my opinion, that is your right answer. Okay, moving on. 46, the diagram below indicates a few of the many varieties of domestic dogs thought to have originated from wolves that were domesticated thousands of years ago. You see a wolf, there's a bulldog, there's collie, there's how do you say this? That Dashun. You know what? I'd like to check it out. I'd like to find out how to say this. So let's see. I always do this whenever I come up upon something I don't really know how to say. I'm going to find out. So let's just put definition and let's just hear it.
Dachshund.
Say it again.
Dachshund.
Ducks hunt. Ducks hunt. Let's do it again.
Dachshund.
It's like ducks in a row. Hund. Ducks hunt. Okay, now I know. Thank you, Google Dictionary. Ducks hunt. So anyway, now we have this bulldog, collie, and dachshund. Hunt. These three dogs came out of wolf, okay, originated from the wolf. The many varieties of domesticated dogs were most likely produced as a result of cloning dogs with desirable traits. Well, kind of looks um, right to me because people have always tried to produce dogs, right, with the desirable traits. However, I have a problem with the word cloning. Cloning literally means replicating the same thing. So, how can you clone something with desirable traits? I mean, if it's the same thing. So D is, it's confusing. A, mutating the body cells of the dogs. Okay. Um, well, mutating could mean a lot of things. Obviously altering DNA, um, selective breeding over many generations. Now that's perfect because that's what actually humans did and are doing, will be doing for a long time. Genetic engineering with specific enzymes. That's for bacteria. That's mostly for cells producing some sort of substance that we need, not necessarily dogs. If you wanted your dog to produce golden eggs, that would probably turn into genetic engineering, but we're not doing that here. We're breeding these dogs over many generations. So B would be my answer, and yeah, I would definitely put my money on B. Okay, moving on. 47. The process shown below is used to. All right, you got bacterial DNA that is also called enzyme. You have human insulin gene. You put them together and is used to determine if a person has a genetic disease. Why would you take bacterial DNA and insulin? And no, A is not correct. Produce human growth hormone. Not correct. Insulin is not growth hormone. Identify the father of a newborn. That's irrelevant. D, produce a hormone to regulate blood sugar, which is insulin. Duh. That is our answer. Do we have any more? Okay, we got, I think this is our last page. And let's take a look at this. A technique used to alter cells is represented in the diagram below. Now, what do we see? We see a body cell. There's a nucleus removed. So you remove the nucleus, okay? You turn literally a eukaryote into prokaryote almost. And then there's an egg cell. Again, nucleus removed. Oh, so there's a body cell and there's an egg cell. Okay, so you take the nucleus from a body cell and you insert into an egg cell. You replace the egg cell's nucleus with the body cell's nucleus, right? Nucleus is fused with the egg cell. Okay, then cell divides and you have new cells. A technique used to alter cells is represented in the diagram below. Wow, that's interesting. I have no clue what's happening here. So I'm going to learn with you. The genetic material contained in the nucleus of each of the new cells is most likely, okay, is most likely identical to that in the original body cell. Okay, identical to that in the original egg cell. Now, it's not going to be identical to that in the original egg cell because the original egg cell nucleus was removed, and so was the DNA with that. So B is impossible. C, 50% of the same as the original egg cell, and 50, again, zero. Zero will be the same of the original egg cell, okay? So C and D are incorrect. A would be the only correct answer because the only thing that's identical is going to be in that new cell, um, which is going to be original to the body cell. 49, scientists have been investigating a way to recreate extinct species such as the saber-toothed cat. Illustrated below. I don't know why, but anyway, moving on. Which technique would use DNA? I say I don't know why because I have a cat who is already causing damage to my hands and arms and whatnot when he tries to play with me. So I don't need a saber-toothed cat now roaming around. But anyway, which technique would use DNA from an extinct species to recreate an organism of the species? Okay. Um, Cloning. Wow. Cloning. I like that because if you want something exactly the same, you definitely want to clone that. Natural selection is not going to work. Differentiation. That is another word for specialization. No, that's for like blood cells or cells, yeah. But mm, selective breeding. Well, my cat came from this cat, so that was already selective breeding. If anyone did anything about it, but I think in this case it's C. It's just cloning. If you want a saber-tooth tiger like that, then you definitely want to clone that. That would be it. All right, guys. Um, this was great. Thank you. I've learned a lot. I hope you did too. If you have any questions, please use the comment area to comment. Okay. Thank you.